welcome everyone to our seminar in precision medicine. I'm Gary Miller. I'm the Vice Dean of Research in the School of Public Health and involved with the precision medicine resource within the CTSA. And just a reminder that our seminar in precision medicine is a collaboration among many groups that are doing precision medicine across the university, including the Precision Medicine Initiative. So it's my pleasure today to introduce Dr. Steve Hyman, who's going to talk about precision psychiatry. I think he's very well known in the neuroscience community. He spent a few years as director of NIMH, and after that became the provost at Harvard for a decade. And then he's now gone back to being a faculty member and scientist and physician since that time. So he's done a lot of really fantastic work in the field of psychiatry, and we are pleased to have him talk about this. So with that, I'll turn it over to you, Steve.
Great. Thank you very much. It's a pleasure to be here. Precision psychiatry, I subtitled it, What Will It Take? Because, in truth, if we're being clear-eyed about it, it is an aspiration, but by the same token, I think we do have the tools to begin to make progress. These are just my declarations of outside interests. Okay, so the problem. So there are some bright spots, some new drugs like ketamine and brexanolone that have been approved, and presumably the M1, M4 antipsychotic drugs. But even these are rather old drugs. Xanomeline has been around, for example, for a really long time, even though it has been cleverly repurposed in a way that blocks its side effects and allows it to be used as an antipsychotic. The problem for psychiatry is that, in truth, treatment discovery has stalled. If you look at this list of prototypes, for the most part, we're dealing with widely used drugs that come from early 1950s prototypes in the case of lithium and antipsychotics, and later 1950s prototypes in the case of antidepressants. And what's more, despite some of these bright spots that I mentioned, like some interest in revitalizing drugs like xanomeline, the pharmaceutical companies have, for the most part, abandoned psychiatric research. And this is despite high unmet medical need, the lack of any treatments for, you can see at the bottom, the disabling cognitive and deficit symptoms of schizophrenia. You probably may have seen in the New York Times some weeks ago the nth article about the maltreatment of people with psychotic disorders at Rikers Island, but also the fact that these people are hard to treat and are not getting treatment. There are no treatments for the core social communication deficits of autism. When I ask people in industry, what would it take for them to return, not yet what would it take to have precision psychiatry, but what would it take to bring them back? They generally say that it is biomarkers, that right now clinical trials using current diagnostic criteria in psychiatry provide highly heterogeneous groups that make it very, very difficult to do clinical trials. And I'll come back to this in a second. So why has it been so slow? Well, not to get us off the hook, but the complexity of the human brain, of course, makes it arguably the most difficult object in the history of science, but it's also inaccessible in life, right? So here's a dividing cancer cell, which reminds us that cancer and other organ pathologies admit of biopsy. We don't have access, except in very rare circumstances, we don't have access to diseased human brain tissue, and certainly not for psychiatric disorders. And this old skull, this is actually an Inca skull with a trephination hole. Well, this is just not the way we can operate, either ethically or medically. Secondly, psychiatric disorders don't have obvious actionable biochemical pathology like A beta or tau or alpha-synuclein. And those clues certainly, while they don't gift wrap answers to how to treat disorders, they are very, very helpful. The genetics of psychiatric disorders, which I will come back to, is extremely complex. Animal models are hard in all of medicine, but in psychiatry, I think they've been particularly misused, and tragically so, where things like the forced swim test, which was developed as a so-so assay to screen antidepressant drugs in some people's minds, have become a model of depression. It certainly is not that. Now, of course, we need cellular models with human genomes, and those cellular models don't have brains, so we need laboratory animals. But they are models of mechanisms, not of psychiatric disorders. And then back to the point of without biomarkers, first of all, human brain heterogeneity is extraordinary. There are nearly 8 billion people on Earth, and everybody's brains are somewhat different from each other. And then the disorders, especially as now diagnosed, are mechanistically heterogeneous, which reflects the polygenic nature of risk and the unutterably complex nature of the exposomes that might convert genetic risk into disease. And this, of course, complicates translational and clinical research. This brain heterogeneity has been, I think, really under investigated or not thought about deeply enough. We don't in psychiatry have any real indication for using non-invasive neuroimaging in normal diagnostics, except to rule out a neurologic disorder. And these failures to identify case control differences, even with large consortia that are very well put together, like Enigma doing meta-analyses with thousands of subjects, still fail to find compelling case control differences. Undoubtedly, the heterogeneity, both of brains and of disorders, is the problem. And of course, going back to why industry is skittish about re-entering psychiatry, is that this heterogeneity confounds clinical trials. Nature has made it hard for us, but human efforts have made a hard problem harder. The current nosologies, DSM and ICD, and this is not, I've had something to do with both of them, especially the ICD, and we can't blow up these systems without throwing all of clinical psychiatry into disarray. But from a scientific point of view, they don't capture very well documented and important features of psychiatric disorders, such as the fact that they are dimensional, not categorical. I'll come back to it, but risk alleles are normally distributed in populations, as are symptom clusters, and therefore, rather than arbitrary categories, we really need to do a better job of identifying the dimensional nature of psychiatric disorders. And then for any dimensional condition, whether it's lipid levels or hypertension, then we need to set thresholds based on data instead of making them implicit in arbitrary categories. This certainly doesn't help clinical trials design. The other thing that happens with these artificial categorical separations is that even though the correlation genetically between schizophrenia and bipolar 1 disorder is 0.7 in terms of common variants, and the first high-impact rare variant for bipolar disorder that was discovered, ACAP11, only reached significance when you included schizophrenia patients as well. But there's remarkably little research on the whole psychosis spectrum. Typically, bipolar disorder and schizophrenia research are segregated, which also doesn't help us reach precision psychiatry, where things are divided up perhaps in a more natural way. There's a lot of interest now in so-called cross-disorder research. I think the main benefit of this, of course, is that it might bring together things like schizophrenia and bipolar disorder, anxiety and depression, and so forth. But the problem is that cross-disorder thinking has to be on top of these problematic. What makes it cross-disorder is that you have to cross the boundaries of these problematically drawn categories. I guess one other and useful side effect, not to be too cynical, of cross-disorder research is that it does generate more papers, because first people do the genetics of schizophrenia and bipolar disorder, and then they can write a paper saying that that there's genetic correlation. But I think ultimately we have to do better than that. Okay, so lack of biomarkers and its impact on treatment discovery and development. I'll show this example. This is a paper that was largely that has authors from the FDA and has FDA patient-level data. And it's a complicated slide, but basically what it says is these are this is in the yellow placebo responses, and in the purple are drug minus placebo. But of course, the two occlude each other and intersect. And without going into the details, I've given you the reference for the paper. Basically, what the FDA analysts concluded, along with other authors, is that if you look at the registration trials for all antidepressants that had been approved, so these are drugs that ultimately all made it over the finish line, the drug placebo difference that is absolutely convincing was only in 15% of patients, and another 50%, of course, got better based on Hamilton ratings, not a great rating scale, but it's what we have. Another 50% got better, but you really couldn't tell whether it was drug or placebo, or more likely, or some combination, and the rest didn't get better at all. Now, the drugs, I think we've come to the view that, we're happy to have them. They are not as effective as we hoped, even when we listen very closely to Prozac, to take that old title from Peter Kramer. But the problem in this data and in these clinical trials is that we don't know ahead of time which patients are likely to respond. Here, the patients who responded at the highest rate tended to be the most severe, which is actually a good thing. But companies are skittish because they have to have very large clinical trials. And in the case of antidepressants, almost every drug that's been approved also included at least one failed clinical trial, one where, say, the placebo rate occluded the rate of drug response. So this is, if anything, this slide is both an argument for the need to come up with better, more efficacious drugs, but it tells us how important it is to have biomarkers, and not only to attract industry, but so that we can do drug development. Now, a quick side note, the idea of precision psychiatry has also entered the world of neuromodulation, both repetitive transcranial magnetic stimulation, which I won't show you a slide of, but there are some interesting new protocols based on iterative use of brain imaging to improve the efficacy of RTMS, but a really interesting, very early case for deep brain stimulation. We'll stick with depression, and this is treatment-resistant depression. So in 2005, Helen Mayberg first published. She was a pioneer in deep brain stimulation for treatment-resistant depression. These are people who've generally not benefited from either drugs or CBT, and in many cases from ECT as well. And based on what antecedent data there was about different brain regions, she turned her attention to this subgenual anterior cingulate gyrus, which is shown here, abbreviated CG25, Brodmann area 25, which is a junction for lots of cognitive, emotional, and somatic innervation. So it was plausible. And all of her patients got a DBS with an electrode stimulating CG25. This is looking at it projected on a human brain, not a cartoon. And in her hands and the hands of her neurosurgical colleagues, about half of the patients got substantially better despite having not responded to anything else. However, double-blind randomized trials of this single region and another region that had been experimented with by other groups in the ventral capsule, ventral striatum. Actually, both trials were terminated early for futility. Now, there were issues, right? Patient selection, but you know, again, without biomarkers, patient selection relies on pretty questionable interviews aimed at heterogeneous DSM diagnoses and maybe physician gut feelings, which is not the way to have a good clinical trial design. And then maybe lead placement. Now, here's an alternative. This is, I think this is a group at UCSF, they're up to N of 2 or 3 patients. But the idea here, which is very interesting in the context of our topic today, is to find a biomarker. In this case, it was activation in the amygdala when people were having their severe symptoms. And it was a biomarker that was considered useful because when they were well or improved, the activity in the amygdala went down. And then, and again, I've given you the reference because I don't want to take too much time on this slide. But the idea is that they could then stimulate various brain circuits to optimize against the biomarker, and found in, this is their reporting on their very first case. So again, we shouldn't make too much of it, as interesting as it is. A 36-year-old woman with severe childhood onset depression who was highly treatment resistant, who got better. But the point is, choosing in this case whether to use the ventral capsule or CG25 or some other area is driven at least by this objective biomarker and allows potentially greater success based on personalization, whereas giving everybody the same site for stimulation, at least in large-scale clinical trials, led to futility. Okay, I'm going to go back to drugs here, but it's just to say that the idea of finding biomarkers that permit treatment development with greater precision is not limited to drug research. So in some ways, I think an important model for psychiatry comes from neurology and from the treatment of Alzheimer's disease. So I think although there have now been more recent successes with aducanumab perhaps and lecanemab, nobody really thinks that these monoclonal antibodies against amyloid peptides are the answer or even all that good, but they are a real beginning. But what's more is that it's been demonstrated that you can do sensible clinical trials for Alzheimer's disease. Now, a bit more than a decade ago, in the first set of clinical trials, when there were no biomarkers, when the patients came to autopsy, which, these were elderly people with a dementing disorder, when they came to autopsy, it turned out that even though the selection of patients for these clinical trials was done by experts in dementia, fully 30% of the patients never had amyloid in their brain. So they never had Alzheimer's disease. Typically, they had Lewy body dementia, but there's also more recently recognized TDP-43 was not recognized at that time, and vascular dementia. The first biomarkers for Alzheimer's were looking at levels of A-beta peptides in the cerebrospinal fluid, and actually, as the disease progressed, A-beta levels shown here in red, not the directionality, but the utility as a biomarker over time. A-beta levels actually decline in the cerebrospinal fluid as they're being deposited in plaques in the brain. But the other toxic protein in Alzheimer's disease and other neurodegenerative disorders, phospho-tau, increases in the CSF. And it turned out you could measure these reliably, and now many other biomarkers, including neurofilament light chain and potentially soluble TREM2. But you could measure these in the cerebrospinal fluid and actually predict who actually had an amyloid disease, who had Alzheimer's disease. And in addition, you would predict that they were going to get sick with it before they had cognitive symptoms. And I think we all are of the view that treating the neurodegenerative disorders early, actually before symptoms, is critical, and the same may very well be true for psychiatric disorders. I think the other thing that's really important is they sensibly started with CSF, and this is a comment that foreshadows what we're trying to do now in the schizophrenia spectrum. They started in CSF. This is, after all, the fluid that bathes the brain. And it is an empirical question which analytes that you might measure equilibrate across the blood-brain barrier and give you reasonable values that you can measure in blood, which is obviously much more useful in larger clinical settings than CSF. And in other cases, they could be developed into PET ligands and PET tests. And in fact, for Alzheimer's now, there will be blood tests for A beta, for pathologic forms of tau, certain phospho-tau species, and for neurofilament light chain, which is a non-specific neural injury marker, which is also useful for ALS and perhaps for head injury. And there are PET scans for, as you probably know, for amyloid and tau. And indeed, not only were these critical in motivating and permitting clinical trials to occur, both in the recent past for these antibodies, but also in the future, but right now, probably for reimbursement to get, let's say, lecanemab, people will need either a PET scan or a lumbar puncture, pending the time when the blood tests are fully validated. But if you think the DSM still describes dementia as major neurocognitive disorder, and that's a sort of a bit embarrassing. It's like saying, well, clinicians in their office want to make a diagnosis, but they could order a PET scan or a lumbar puncture. And this is a sort of a return to the pre-modern era when clinical experts, we already know, could not actually distinguish the various types of dementia, and this has treatment implications. And we really ought to move on from these kinds of descriptions. Okay, so the DSM. So the DSM, or the very similar ICD system, but the DSM is the way we're making diagnoses today. And you can already tell that I don't think these are helpful for clinical research or clinical trials or the future of what we aspire to with greater precision in psychiatry. What's important is that the intellectual foundations were set in the 1960s by people like Robbins and Gouset and their colleagues at Washington University in St. Louis. They don't deserve criticism, but rather praise. They were pioneers who recognized that there were not appropriate laboratory or other objective tests. And so they I think, understood themselves to be so-called descriptive psychiatrists. But they had to be that because when they set these intellectual foundations, there was actually, in the 1960s, there was no, I don't think there was any neuroscience graduate program yet anywhere in the world. The first graduate programs sort of coalesced around 1970. There was clearly no modern genetics. There were just mostly Mendelian ideas, although there actually was already a paper from Gottesman and Shields that argued that schizophrenia was polygenic. It's just, I don't think that the descriptive psychiatrists could read that paper. It was written in a very different language of quantitative genetics, but I guess they could have read the abstract and the discussion. But at any rate, they treated psychiatric disorders almost as if they were Mendelian or natural kinds. And of course, it was before any kind of data science or computing. And it's unfortunate that the system we have today is really based in that pre-modern era. But at any rate, they being honest, they said, we don't have this information, but we predict that clinical descriptions, especially clinical descriptions where symptom complexes co-varied, they hung together across patients, would converge with laboratory studies. They would turn out to be categorically separate. They got this from Kraepelin's attempt to separate schizophrenia from bipolar disorder. Although Ken Kendler says that they were more Kraepelinian than Kraepelin. Kraepelin always had an appropriate doubt that he really had separated the two. Course of illness, positing lifetime stability of diagnosis, which has turned out not to be the case, and family history. So a sort of a Mendelian view that these disorders would breed true, that if you had a parent with schizophrenia, the offspring would have schizophrenia. And that has also turned out not to be true. Again, it would have been nice had they read Gottesman and Shields and understood it, because polygenic disorders would not be expected to breed true, as this highly complex panoply of risk alleles will segregate very differently in forming different gametes. So this idea of convergence has been refuted. And as I've said, psychiatric disorders are polygenic. Our brains are not at all like Mendel's peas, and thus we shouldn't expect these disorder phenotypes to breed true. It's interesting, when I was early in my training and research career, there were people who were writing papers finding bipolar patients in so-called schizophrenia pedigrees. There was a lot of doubt, and I think people worked hard not to see these anomalies, because there was a lot of cognitive straitjacketing, I might call it, based on this strongly held theory that treated these disorders potentially as natural kinds. But in fact, affected individuals with any psychiatric condition, actually with any heterogeneous, complex, chronic human disease, have genetic loading with a unique grab bag, and in the case of schizophrenia or bipolar, from literally among thousands of risk alleles that have loading above a threshold, but the threshold's not a bright line. And then they have bad luck, meaning during development, maybe they have unfortunate leaky transcriptional control, or they have unfortunate somatic mutations, or who knows what, and then environmental exposures that gives them disease. So the point is that this leads to enormous heterogeneity, but at meiosis with the genetic assortment, it leads to offspring who do not follow the model that either Kraepelin or the St. Louis school imagined they would see. The heterogeneity I've already talked about. The whole basic premise for precision medicine is that outcomes are not equivalent for people with the same diagnoses. And the point of biomarkers ultimately is not going to be to make diagnoses per se, but actually even to stratify within them so that we can match people with the right treatments. It's just that in psychiatry, we're at a much earlier level. And as I mentioned at the outset, the heterogeneity of these disorders as we now diagnose them is such that imaging is not yet clinically indicated except to rule out a neurologic disorder. And so I suspect that biology will be of great aid to people studying doing non-invasive neuroimaging to MRI studies, by providing ultimately, if this all succeeds, independent biological markers, right? Biological markers that are orthogonal to the MRI studies that will help divide people into sensible, more homogeneous groupings where imaging has a much better chance. Now, this is not, it's really important that MRI and functional MRI have worked beautifully in cognitive neuroscience, where people can control enough of the variables, and have been used iteratively in an individual kind of way for RTMS in the Stanford-based Sync protocol. But it is just the fact that defeated by this heterogeneity and without independent markers, we can't separate cases and controls by imaging. And of course, that goes along with the problems we saw in clinical trials. Now, the other problem with these, with the categorical nature of the DSM based, and now look, I mean, Robbins and Gazet had the right humility. They said, we don't have laboratory tests, we don't have this, we're going to predict that they will converge. But they did make what we would now call a bad decision by treating they and their followers, and especially Spitzer et al., I'm afraid to say at Columbia, in formulating the DSM and choosing to treat them as categorical disorders at a time when it was already clear that most common chronic human disorders, type 2 diabetes, for example, atherosclerotic heart disease, were better understood dimensionally. And in fact, now we can see that these polygenic risk alleles are normally distributed in populations with no bright line cutoff. They are shared among what we now consider to be separate categorical diseases, and many symptom complexes. Just think about ADHD or major depression. Symptoms are also widely distributed. I don't know if they're normally distributed, they're probably tails, but they're distributed in populations. And so the idea that these could be captured categorically is really defies one's imagination how one could get there. But the point is that there are no bright lines that separate illness from health or disorders from each other. This clearly has to be a confound in reaching some kind of precision, better psychiatry. MDD, as now diagnosed, would allow you to include a 9-year-old girl with familial loading for depression and a 68-year-old male who's just getting a diagnosis of Alzheimer's disease or mild cognitive impairment. Pathophysiologically, they're not the same. They won't likely respond to the same treatments. It's really time that we readdress these things. Now, the idea of that longitudinal course, and this is a Kraepelinian idea, would help delineate one disorder from another, also really falls apart. This is a slide I got from Arno Pelotti in Finland, where we have a 10,000 person psychosis spectrum cohort called SUPER. And they've done these very nice charts from the electronic medical records here, just calling out diagnoses. But I have to say that my experience as a clinician early in my career was very much this, that if a young person showed up, for example, with a florid acute psychosis, I had no idea what the ultimate diagnosis would be, and it might have been months or years before we figured it out. And if you looked in the chart, you would see not diagnostic stability fitting this Kraepelinian categorical model, but something much more like this with diagnostic instability. Now, the problems with this very typical clinical pattern are many, but one that I want to point out is that you could have recruited this young person at the age of 16 and a half into an imaging study or any other clinical study of MDD, of major depressive disorder. And then at 25 Or you could have recruited this person into a study of bipolar disorder, and then at 32, the final diagnosis is schizophrenia. So and this is not to blame diagnostic categories, this is just to say without any kind of objective biomarkers that look at pathophysiology, we really introduce all kinds of noise into our clinical studies. So no wonder we don't have good case control studies with imaging. Now, the other thing which is important is that when we treat these as categorical, as the DSM does, these successive diagnoses become comorbidity, right? As if the person has, in this chart, 6 different diagnoses. Actually, I pruned this tree that this person would have had many other diagnoses. They can be successive with each other, or they can coexist, as alcohol and substance use disorder and conduct disorder coexisted with adjustment disorder before this person got a bipolar diagnosis. In fact, this is very likely to be, now this is a hypothesis, I don't know this, but very likely to be a single pathogenic process. After all, this person's genetic sequences are not changing, the person's brain is developing, the exposures are changing, true, but there's probably a single pathogenic process which is having different forms of expression over time in this person. And if we're going to have precision psychiatry, where we perhaps at the age of 15, we could have come up with a better definitive treatment. And again, in my clinical experience, people who ended up with a final diagnosis, say, of bipolar disorder, often had a treatment trial with lithium or an anticonvulsant delayed by months or years as their diagnoses shifted and shifted and shifted. So this is one of the problems that we have to solve. Not insuperable if we can use the genetics and the neurobiology to tell us where to start to look for biomarkers. Okay, so the genetics. I'm just going to use this Manhattan plot to remind us of how complex the genetics is. This is the Psychiatric Genomics Consortium Schizophrenia GWAS Wave 3. Our goal at the Stanley Center, working with collaborators around the world, is to reach 250,000 cases of schizophrenia. It's a bit of an arbitrary number, but the idea is not to saturate the number of variants, which is not all that useful, but to saturate the new biological information, and then we ought to stop. But it just again, if you have thousands of alleles segregating, you would expect the kind of intrinsic heterogeneity. Even though these are going to coalesce into a finite number of pathways, it is still an issue in front of us. We know that these loci actually are very hard to decipher in terms of biology, because they're chunks of chromosome with tens or hundreds of single nucleotide polymorphisms and linkage disequilibrium. But what we can do, and now look, this is a first step toward a biomarker or a biomarker-ish, and that is to turn these loci into polygenic scores. So basically, these are a weighted sum of all of the risk loci in the genome, and then for an individual, you can compare their polygenic score. So a weighted sum, weight is by the effect size of the allele of all of their risk alleles, percentiled against an appropriate population control. And a point I'll make in a second is that if we're ever going to use polygenic scores in an equitable way, we have to recognize that because of genetic drift that changes patterns of linkage disequilibrium, when we have a study like the one I showed you here that is almost entirely of European ancestry, we can come up with polygenic scores for individuals who have European ancestry, and they degrade with genetic distance. But I will come back to that in a second. What matters is that this at least begins to stratify individuals. We can't make good individual predictions. These are really probabilistic and also partial because people are working on it, but they don't yet include rare variants. But what we can say for a lot of polygenic scores is whether somebody has substantially less risk or more risk than the average in their population. And I'm going to skip this one. This is work of Alicia Martin. She has galvanized people to recognize that polygenic scores. So this is a synthetic score from biobanks, but score performs less well in people with indigenous American genetics and less well in South Asians and even less well in East Asians. They perform terribly in African populations, which have the highest genetic complexity of any human populations because they're the oldest, where humans are from. And so if we're ever going to use polygenic scores, even as a little bit of a piece of precision psychiatry, we had better do our homework and globalize human genetics, or else we will be, in Alicia's words, increasing rather than decreasing health disparities. Right now, Europeans, who are about I don't know, 15 or 16% of global populations, represent still around 90% of all human genetics. And frankly, if you look at collections of things like iPSCs and other biospecimens, it's even worse. All right, so TJ is, I talked to earlier today. TJ, when he was in Mark Daly's group and is now an independent investigator here at Columbia, using whole exome sequencing, found these higher impact loss of function variants. And I don't want to go into the detailed biology here. I'll come back to the fact, though, that a lot of both these high impact variants and the GWAS hits that we think we have interpreted biologically are pointing their fingers at synapses, at their structure, at their function, at their plasticity, which will be very important for understanding biology and potentially for biomarkers. But I wanted to introduce this to show that in addition to these common variants that we see in polygenic scores, there are both high impact rare variants, but there are lots and lots of rare variants that don't have such impact, that we have not yet integrated into such predictors. Even today, I guess if you had a clinical trial that was made up of a studied population, if there were some people who really had no benefit, even today, doing a polygenic score wouldn't be that informative, but it could tell you whether those people were even in the ballpark or whether they were fully misclassified, you know. So if these were the utter failures of an MDD trial, maybe polygenic scores could help you explain why a trial, or at least certain patients, didn't respond. That may not yet be true in psychiatry. It probably is true in some other areas like cardiovascular medicine. Now, another issue that impacts on the possibility of precision psychiatry and does impact on this idea of what really are the disorders and should they be categorical, which again, I've already alluded to, is if you look here between schizophrenia and bipolar, the correlation in common variant space is 0.7. And between bipolar and MDD, there's correlation. between MDD and anxiety disorders. There's enormous correlation. And so the question really is, do we have separate disorders that we're finding correlation in, or have we really just thought about it wrong? It's even I'm showing this to show that it's not just that these alleles are shared among disorders, but these alleles are shared with normal, even desirable phenotypes. So polygenic forms of ASDs, OCD, and anorexia nervosa, which I didn't circle, I should have, share alleles with years of education, so-called educational attainment, and also with grades in college. So I guess in some ways the good news is that these diagnoses are not pure deficit. They bring with them also some strengths. But again, when we're thinking about precision, we should be thinking about safety as well, right? And what these genetic correlations are telling us is that if we target the wrong alleles, we might be actually impairing somebody's cognitive ability while we think we're treating their OCD or anorexia nervosa. So for treatment development, we have a much longer way to go on this and understanding what these alleles are doing. But it is a kind of a warning flag. We already have drugs that probably are bad for people's cognitive development. Phenobarb is one. There are many low-income countries where it's the only anticonvulsant in the formulary. And kids who get phenobarb are getting their brains bathed in an inhibitory, increasing GABA levels, which is good for blocking seizures, but not very good for learning in school or for brain plasticity and development. And of course, we have no idea what the relevant alleles are here, but again, it's as we get into precision medicine, precision psychiatry, which of course we all aspire to, there are important safety warnings as well. It's not just about efficacy. So to summarize this bit, let me put it in a different way than I have already, which is people will often say to me, why are you critical of the DSM? It's giving you all this information from GWAS. And my answer is, so might other classifications, right? These alleles are widespread across both healthy and pathogenic and pathological phenotypes. There's maybe nothing special about DSM MDD or DSM schizophrenia. And in fact, the fact that there are these high genetic correlations may not really tell us that everything is succeeding. It may tell us that we've divided things up wrong. And in fact, one of the reasons I showed you the Schema Consortium high impact exome sequencing results that TJ was the first author on is to remind me to tell you that the first such high impact protein disrupting mutation found for bipolar disorder, ACAP11, only became significant when schizophrenia cohorts were included. So not only is there sharing between schizophrenia and bipolar disorder among common variants, but N of 1, take it with a grain of salt, but the very first high impact protein disrupting variant is also shared between schizophrenia and bipolar disorder. So the question really is, to what degree should we be talking about the pleiotropic actions of pathogenic alleles? And to what degree is this simply an artifact of our diagnostic system? I guess there's an opportunity to write a lot more papers, but I think as we think about how we want to stratify patients, because we'll agree that people with in the tails, people with Kraepelinian style affectless schizophrenia and people with bipolar type 1 are phenotypically different. They deserve different treatments, but how we actually divide it up to have precision psychiatry is in question. So at the Stanley Center, we have this 10,000 person cohort from Finland against the background of FinGen, which has 500,000, so 10% of Finland's population subjects. I'll come back to it in a second, with access to their electronic records, medical information, even early cognitive testing, their APGAR scores, blood samples. So for this psychosis cohort, we have at least GWAS and whole exome sequencing for all samples, and for nearly 1,000, we have whole genome sequencing. Still too expensive to do whole genome sequencing on all 10,000, but I think we have good information from the GWAS plus whole exome sequencing. 87% of participants have consented for iPSC research and recontact, and our colleague Olli Petolainen has already actually collected about 6,000 samples. So we have frozen PBMCs to make iPSCs when we have an interesting phenotype or genotype. It's too expensive to come up with 9,000 or 10,000 iPSC lines right now. All right, so at least for the rare variants that are of high impact, we know everybody in this super collection who has a psychotic disorder and all of these different mutations that are protein disrupting. We also know, frankly, carriers who do not have a psychotic disorder. And for most, we're busily collecting cell lines so we can look at their iPSCs. And we're about to figure out whether it is timely to recontact them for rephenotyping. And sadly, that's a really open question, because I always ask, if we go back and we've already done good cognitive testing and all the things you would normally do, and do we know how to phenotype patients any better than Emil Kraepelin would have? Or should we wait and, we can't wait too long. We want to keep people engaged in this study, but, do we really know what we want to get? Well, there may be, by getting biological fluids, we don't have to know the answers now. We can biobank them. But I think it is a really important question, again, as we aim for precision psychiatry, and we have this most valuable of all resources, which are recontactable cohorts. What are we going to ask them? We can't keep bothering them. I think we really have one more bite at the apple, so we better know what we're doing. But this is just to say that we are collecting these cells, and we're already using some. Now, this again goes to this topic that I'm focused on here, and we're getting near the end of the talk, which is this is ICD nomenclature, not DSM, but they're very similar. We collected, at my insistence, but with the agreement of the Finns, psychosis spectrum, not just schizophrenia, not just bipolar. And there are these diagnoses that are, I feel, completely made up, like schizoaffective disorder, of course, other psychosis or psychosis NOS, and then there's schizophreniform disorder in the DSM. Depression with psychosis, I think, is at least a better capturing something more likely to be real. But I really think that we should be trying to use these kinds of cohorts. We should be collecting not schizophrenia cohorts or bipolar cohorts, but psychosis spectrum cohorts, or internalizing disorder cohorts, if you accept that nomenclature, and reanalyzing them from the bottom up. There are more patients with schizophrenia because they're sicker. They tend to be in care. We can find them, but we actually don't know, all that much about what is going on in all of these patients. And I think that maybe the best thing to do right now is to think of this as a spectrum with the classic, as I called it, Kraepelinian schizophrenia, in one tail and bipolar type 1 in another tail, do the genetics, do the phenotyping, get whatever fluids we can for biomarkers. And insofar as we can, this is just the very beginnings. I don't want to overpromise, but really recreate these phenotypes from the bottom up. And that's what I have in this slide. This is my straw person reconceptualization, which is to begin with the psychosis spectrum and reanalyze from the bottom up. I first wrote about this in 2005 in an article in Nature Neuroscience. It was not at all realistic then. I think it is on the borders of being realistic now. Okay, we have only a few minutes, so I am going to, and I was too ambitious, so I'm going to skip ahead to the idea that began in 1983 with the Feinberg hypothesis, that given the adolescent time of onset of schizophrenia and what was then known to be postmortem from Huttenlocher, that people have the largest number of synapses at ages 10 or 11, and then they go through a period in adolescence of synaptic reorganization as you develop a mature brain. But there's net synaptic pruning. You lose about 15% of your synapses during this period of brain maturation. Strong synapses are strengthened. This is the Hebbian neurons that fire together, wire together. Weak and unused synapses tend to be pruned away by microglia. And I did have this picture of microglia, doing their thing here. I don't know if I can show the video. It's like a horror movie watching them chomp away at synapses. Since then, imaging, this is Ty Cannon's work, has shown cortical thinning, and he thinks he can see that people who go on to have schizophrenia have greater and faster thinning in prefrontal and temporal regions. Not everybody is able to capture this, but it is credible because there is convergent evidence from postmortem studies that finds pretty much the same thing. So the genetics of schizophrenia finds not only a lot of synaptic proteins, both in rare variant space and common variant space. Probably about a third of the genes that have been implicated are encode synaptic proteins. And then this interesting finding that complement factor C4 is the largest of the common variants associated with schizophrenia. You don't see this high peak in East Asian populations, interestingly, because of patterns of linkage to equilibrium, but C4 is still a positive finding there. And then this is the work of Steve McCarroll and his graduate student Ashwin Sarkar, who basically mapped this locus, which is in the on chromosome 6, and found that the odds of having schizophrenia went up as you had more copies of this is actually a structural variant in the genome, more copies of complement factor C4. There are different versions of it. Humans have C4a and C4b. It is C4a that is the risk gene. Interestingly, the highest risk alleles give you the highest levels of RNA expression and also protein expression of C4. And this is just, these are just brains of schizophrenia in control. Even normalizing for genotypes, and it's important. This tells you that genes by themselves don't do all the work. The environment and other things matter, because even correcting for genotype, people with schizophrenia have higher levels of C4 in their brains than controls on average. There's a lot of variation. And by the way, this is, rather than being disappointing, this gives you an opportunity for stratification. I mean, did the people with less C4 have less synapse loss or less cognitive degradation? I don't know, but I mean, these are the kinds of things that have to be followed up on. This is just saying that when you knock out C4 in mice, you get an increased number of expression. And if you overexpress human C4a in mice, you overprune. So given this, we've just begun a biomarker study. There are eight institutions involved. The pilot sites are Penn, Sinai, and Yale. And the key thing here is that we do get cerebrospinal fluid every 6 months. People told us that would be impossible, but actually, while it's hard to get people into their first appointment once they've had a lumbar puncture from a very able neurologist, each of these sites has almost lumbar puncture clinics. Takes about 5 minutes. People have come back and every 6 months, and we've had some 2-year returnees, and along with CSF, we're getting blood and plasma and DNA, and we're freezing cells, and we're doing a standardized cognitive test that Raquel Gur generated and symptomatic tests. We're doing also low burden structural imaging and some resting state. I suppose we might see differences in cortical thickness, but we're trying to keep this because we're getting CSF. We're trying to keep this relatively low patient burden, and so SV2, which is the transporter that transports glutamate into synapses, is actually just sub-threshold among the high-impact genes, the so-called schema genes that TJ Singh has worked on, which is really interesting. But it's also, it turns out, it's the target of a very good anticonvulsant, and it's also been turned into a PET ligand, and it's a presynaptic marker. So now taking this idea that synapses and complement and overpruning might be involved in schizophrenia. So just in Alzheimer's disease, what you can see here in the red is that in a cognitively normal person, there's much more SV2A expression, and it's reduced in Alzheimer's. And we're pursuing this now in schizophrenia. Yale and Penn have very strong PET groups that are doing this. And, this is, the signal will, I'm sure, be muted compared to Alzheimer's, partly because in young people, there's still synaptogenesis, and in Alzheimer's, there's very little synaptogenesis. But already you can see a hint that compared to healthy controls, this is the cingulate gyrus, anterior and posterior. This is prefrontal. Healthy controls have more of this SV2A signal, so presumably more presynaptic terminals than people with schizophrenia. Neural contraction 2 is way down in Alzheimer's. It's way down in schizophrenia. It is a complement inhibitor that's part of the synaptic complex. This is all, by the way, very preliminary data. This is from Paul Worley, who's a collaborator at Hopkins. So the idea here, just to finish up, is that we think that for severe diseases like the psychosis spectrum, that there will be measurable biochemical changes in the brains of people who have them, and that we're not looking for all or nothing, something categorical. We're looking for the ability ultimately to stratify people. And looking again aspirationally, I really don't want to overclaim, but the people who have the greatest synapse loss you might imagine you would treat differently. What I was going to show at the very bottom is there are actually people working on the C3 receptor on microglia, which is only expressed on microglia and peripheral monocytes. So it might be safe, but you wouldn't do this without a biomarker, right? You're not going to mess with brain maturation blind and wait to see what happens to cognition. You need a biomarker. But the point is that this kind of research gives promise through genetics and neurobiology, inspiring us to go after certain biomarkers that could then inspire therapeutic discovery that would give us something like this aspiration of precision psychiatry, or at least better therapeutics. And so let me stop there. Sorry, I've gone 5 minutes or more over. Again, I was a little bit too ambitious, but I don't know if we have time for questions. Let me turn to my host here.
Yeah, so it's fine. If anyone has any questions, if they could use their raise hand feature, that would be great. Yeah, I can start off. I have many things that are very interesting here, but when you are start detecting some of these like potential biomarkers, have you done like any non-targeted or untargeted mass spectrometry on
Yeah,
that?
so
plasma?
yeah, so we are, but we're very concerned about false positives. So what I didn't say is that the ultimate use of these biomarkers would be for clinical trials in industry. So as we develop these assays, we've been transferring them to companies who are supported by the Foundation for the NIH, and one big involvement is right now is from Janssen. But the idea is that we want companies to be confident that the assays in their hands will be bulletproof, and they can use them in clinical trials. And as part of that agreement, the companies are doing this untargeted with different proteomic methods, or well, let's call it hypothesis generation. I don't have any results yet, but it is definitely something that we're doing.
But even like not just the proteomics, but in the small molecule space, because like I always find it intriguing when you have this good biomarker, there's likely hundreds of other things that are changing in that direction. And one of those things might be a more reliable marker.
Yes, absolutely. So we are, what we're going to end up with is a repository, and certainly metabolomics will be done, on this, and other kinds of searches for potential biomarkers. But it is, we're still in the pilot phase, although I have to say it's been more successful in collecting CSF and blood and plasma samples than was predicted. And so we will be able to do those follow-up kinds of assays.
So in looking at some of the earlier things you were showing, like on depressive disorder, do you see any of like the sort of lack of response being due to drug metabolism and like dietary factors that affect that, or?
Yeah, that was the first place people looked. And, there are some people who are slow metabolizers who have a lot of side effects, but these clinical trials failures are not primarily metabolic. People believe they are pharmacodynamic, and that MDD as diagnosis is just way too heterogeneous. And the drugs are actually not that good, but they're good enough that if you had good biomarkers, they would look a whole lot better.
Okay. All right. I'm not seeing any other hands. Any other questions from the audience? People are ready to walk home in the dark and snow here in New York City.
Absolutely.
So if I don't see any other questions, I'll go ahead and end it here. Steve, thank you so much for that.
Thank you.
It was fantastic.
All right.
Exactly what we're hoping for, and hope to have future interactions with you. So thanks again. Thank you very much.
Take care. Bye-bye.